So now we're moving from the overall topic of cell energetics into an area where we're talking about the production of proteins by a cell. And that's going to start with talking about the rough endoplasmic reticulum. So I guess the first obvious question is, what is the rough endoplasmic reticulum? Well, just as a few kind of side notes, facts about it before we really get into it. First off, the rough endoplasmic reticulum takes up somewhere between 5 to 30% of the cellular volume, so it can actually be quite an extensive organelle in the cell. And it's made up of these interconnected flat sheets that are all physically connected to the nucleus. And in addition to that, it has many ribosomes that are attached to those thin sheets. This is actually how it gets its name, the rough endoplasmic reticulum. The attachment of all these ribosomes make it appear to be rough under a microscope. So the rough endoplasmic reticulum is really responsible for protein synthesis and modification. So specifically for proteins for membranes and also certain other organelles, and also export of those proteins away from the cell as well. So these are the types of proteins that tend to be made in the rough endoplasmic reticulum. On the other hand, we also have the smooth endoplasmic reticulum, so named because it lacks those ribosomes that are on the surface. Now this also can make up a fairly sizable chunk of the volume of the cell. Anywhere between 5 to 50% of the cellular volume can be smooth endoplasmic reticulum, depending on which particular cell type that you're talking about. Now, the smooth endoplasmic reticulum has a slightly different morphology, where it is these interconnected tubes and is kind of shaped differently than the rough endoplasmic reticulum. We're not merely talking about rough ER without its ribosomes. The smooth ER actually has its own distinctive morphology to it. And the smooth ER is physically attached to the rough endoplasmic reticulum, and therefore kind of by extension to the nucleus through the rough endoplasmic reticulum. So let's talk about some of the functions of the smooth endoplasmic reticulum. So for instance, the smooth endoplasmic reticulum is involved in lipid synthesis. Some of that happens here, but not all of it. And also metabolism of certain kinds of toxic substances, detoxification. But it's also involved, we're going to come back to this later on in this class, that it is also heavily involved in calcium storage for the cell. On the inside, or in the lumen of the smooth ER, there's high concentrations of calcium, and that can be released at certain times. We've already talked about this specifically when we were talking about calmodulin earlier. So, just in general, the endoplasmic reticulum can extend throughout the cell, and it makes physical contacts with multiple other organelles and the plasma membrane. And this could allow for lipid movement throughout the cell. Remember that the cytosol is an aqueous environment, and therefore lipids are not going to have a really super easy time moving through that, especially in some directed manner. However, the lumen of the endoplasmic reticulum is somewhat more conducive for that, and so this could allow for some of that lipid movement. So let's go ahead and kind of take a look at what this would kind of look like. What we have over here is, this is rough endoplasmic reticulum here. And over here we have some smooth endoplasmic reticulum. And you'll see here in the rough endoplasmic reticulum we have all these little ribosomes that are connected all over the surface of that. We're going to come back and talk more about specifically what those are doing in just a few minutes. But then over here in the smooth endoplasmic reticulum, you'll notice that first off we have a very different shape here with all these tubules that are interconnected, rather than flattened sheets like you have here in the rough endoplasmic reticulum, but we're also lacking the ribosomes. This entire thing would be attached to the nucleus. The nucleus would be sitting like down here, kind of, nucleus, and would be attached to the rough endoplasmic reticulum, kind of along this margin right here. Okay, just kind of give you another sense of this. Here is the nucleus of the cell, and right over here we have rough endoplasmic reticulum, and again we have ribosomes that are scattering the surface of the rough endoplasmic reticulum. So you can kind of get a sense of that there. Okay, so now that we've talked about the organelles, or some of the organelles at least, that are involved in the creation of proteins, let's begin to talk about actually how that works. And particularly, let's talk about protein trafficking, or how do we get proteins, the creation of proteins happening in the areas that we want it to happen. So let's just remember here a little bit about polypeptide origin. So the very beginning of this process is transcription, in which we get DNA that is going to be converted to mRNA, or messenger RNA. This is going to happen in the nucleus. That messenger RNA is then going to leave the nucleus and then head outside the nucleus. We're going to talk about more where this happens. And that mRNA is going to be translated into a polypeptide. And this is going to be happening specifically with the help of ribosomes. So all polypeptides are originating on ribosomes. Now, polypeptide movement, however, is not a random event. Somewhere along this chain, some sorting is going to occur. Polypeptides are going to be going to different places in the cell. And let's talk a little bit about how that can happen. So this gives you a rough kind of overall idea how this can happen. First off, up here at the top, we have ribosomes. Again, remember, all of our polypeptides are starting with ribosomes. But from there, these ribosomes can translate just directly here into the cytosol. Or they could be translating a protein into the rough endoplasmic reticulum, or these ribosomes could occur in the mitochondria, in which case we're making mitochondrial proteins as well. So one of these three places is where it's going to go. From the cytosol, it could then go down and in plant cells become part of the chloroplast. We then direct it back to the nucleus or into peroxisomes, another organelle that we're going to be talking about a little bit more later. In the rough endoplasmic reticulum, however, it could also go to the chloroplast in plant cells, or it could go to the Golgi, which we're going to be talking a little bit more in a lecture after this. From the Golgi, proteins can go to the plasma membrane. They could be secreted out of the cell, or they could end up going to the lysosomes, in which case they could also become secreted after they go to lysosomes. So really, from the origin of being on a ribosome, proteins can end up being directed a lot of different ways. It's not just one simple pathway that they follow. So how in the world does this happen? And one of the ideas of how this happens was known as the signal hypothesis. The signal hypothesis states that the vast majority of proteins are going to have a signal sequence somewhere on the protein, either a signal peptide or some sort of signal patch on a peptide that is going to direct where it's going to go. This is usually at or near the N-terminus of the polypeptide. And then these signal sequences function to direct the protein about where it is supposed to go, or helps guide it, or signal the other cellular machinery to guide it to where it needs to be going. Okay. So let's talk about how this direction would go through that path that we talked about a few moments ago, through the RER, and then on its way there. So let's talk about transport specifically into the RER. So what's going to happen is mRNA and a ribosome are going to meet somewhere in the cytosol. They're going to join up there. So here's a little image of what's happening here. What we have is this is the plasma membrane of the rough endoplasmic reticulum. This is the lumen, or the inside of the endoplasmic reticulum. Up here we have the cytosol, and right here we have some mRNA. That mRNA has been bound already to a ribosome, and that ribosome has begun to translate this into a polypeptide. So that's what this is, polypeptide right there. And what we have right here is the signal patch, indicated in red right here. So what happens at this point? Now that we have this mRNA that has begun to be translated by the ribosome, what happens here? Well, the first thing that happens is that the signal sequence is recognized by this particular other protein called the signal recognition protein. So This has about 6 polypeptides, or has 6 polypeptides, and 1 RNA chain protein component that is GTP binding. And this is going to bind to the signal sequence and the large subunit of the ribosome. And this is going to cause translation to pause where it is. So, right over here, this is our signal recognition protein right here, and this is going to bind over here to the large subunit of the ribosome and also to the signal patch right here. And that's what we have. Here it comes over, it binds to that. From this point now, the signal recognition protein is going to bind to the signal recognition protein receptor, which is a docking protein on the endoplasmic reticulum surface. So now, this is going to bind with the SRP receptor, which is signaled by this right here. That's on the rough endoplasmic reticulum surface. So, boom, there we go. At this point, the signal recognition protein is going to release, and the signal recognition protein receptor is going to move away. At that point, translation is going to resume. So, this whole complex here, the SRP and the SRP receptor move away, and then translation is going to resume into the RER. And that's what we have happening here. This process is called co-translational translocation. Essentially, we're translocating the polypeptide, moving it into the RER as it's being translated. So we are doing these two things at once. We are translating this polypeptide, and we are moving it into the RER lumen at the same time. And this whole complex right here that's moving it in is called the TRANSLOCON. This is an entire complex of proteins that help this process along, and we're going to talk a little bit more about that specifically. So, the first part of the translocon that we're going to talk about is the protein translocator channel. This is a specific channel that the polypeptide is being translated through into the rough endoplasmic reticulum, and it is at least 7 proteins. And the first of those proteins is what's known as Sec61. This is what forms the actual channel. This is a 10-pass protein, so it's actually a fairly large channel, and this is what's allowing the polypeptide to move through the plasma membrane. We also have another called the TRAM protein complex. TRAM coming from the abbreviation here, translocating chain associating membrane protein complex. This is going to bind to the signal sequence and then direct the protein through the protein channel. So it's essentially helping guide the nascent polypeptide through the channel. Next, we have the TRAP complex. The TRAP complex is short for the translocon associated protein complex, and this is going to function to keep the polypeptide from leaving the RER. This keeps it from then moving back out very easily once it is inside. Very conveniently, this acronym TRAP kind of also describes what it does. It traps it in the RER. Finally, we have the signal peptidase complex. This is going to cut the signal sequence off of certain polypeptides once they're in the rough endoplasmic reticulum lumen. Once it's been moved into the rough RER or the rough ER, we don't actually need, for many proteins, that signal patch anymore. So we can just go ahead and cleave it off and presume as normal. Now, there are some proteins that have some further direction that needs to happen to it, so they retain that signal patch after they're already in the rough endoplasmic reticulum, but for most of them, it's going to get chopped off. Then also we have the ribosome receptor. So this is the actual receptor that the ribosome is binding to that actually helps hold it in place whilst translating the polypeptide into the rough endoplasmic reticulum. And there are a lot more. This actually isn't a complete list of all of the parts of the translocon, but I'm just going to leave it with that for now. So, again, this is what we have. So this whole complex here be TRANSLOCON. And again, the only thing I'm really showing here is what would be essentially Sec61, the actual channel going through the membrane. But again, just know that there would be a whole bunch of other proteins that are associated with this, kind of helping the entire process of co-translational translocation on its process. Okay, once the protein has been synthesized, the ribosome is going to unbind. This whole process of actually synthesizing the protein into the rough endoplasmic reticulum is going to take about 2 minutes. So it's actually, in the grand scheme of things, a fairly quick process. Now, that's slow for synthesis reactions, but yeah, it's not, I mean, it doesn't take that long. Now, there's a lot of modification that can happen to the protein once it's in the RER lumen. One of those things is what's known as glutamate carboxylation. So, essentially, we have this enzyme glutamyl carboxylase that takes a glutamate and sticks an extra carboxyl group on there. And it gives the ability to the protein to be able to bind calcium. And much of this is done with this particular glutamate carboxylase that's vitamin K dependent, which is one of the reasons you need vitamin K. Another thing that can happen is glycosylation, which is the addition of carbohydrates to make glycoproteins. For instance, N-link glycosylation or O-link glycosylation, which can happen to serine or a threonine. So essentially here, an aspergine here, and we have stuck a carbohydrate. This is N-linked, therefore. We can also have things such as palmitoylation happening, which is the addition of a palmitate at cysteine on integral and peripheral proteins. This addition of the palmitate, which is a fatty acid, will help to anchor peripheral or integral proteins into the membrane. So this is important for keeping these polypeptides associated with those membranes. You stick a lipid on them, and that lipid then can integrate into the membrane and hold it there. So for instance, this is an example of that. Here's palmitate here, and then we're adding it on here to the cysteine residue, and we get this bond. And then this fatty acid chain here can then go into the membrane. And so again here, this is kind of showing that again. It's really hard to see, but here we have a protein polypeptide, and here's a palmitate right here, and a palmitate right here, and a palmitate right there. That's holding it and keeping it associated with the membrane. Okay, let's talk a little bit now how we have the actual formation of the tertiary and quaternary structure of these proteins. And a certain class of proteins called chaperones are very important for what's going to be happening here. So let's talk about this for a minute. Some of the different things that need to happen during the course of forming the tertiary and quaternary structure is actually forming disulfide bonds that are going to be between cysteine residues. So the SHs, or the sulfur on the side chains of cysteines, can bond to each other and form these tight links between fairly distant places in the polypeptide chain. One of the proteins that's responsible for this is protein disulfide isomerases that will help form this. These enzymes cause formation, breaking, and reformation of disulfide bonds until it's the most thermodynamically stable conformation is obtained. So it just keeps forming them, reforming them, breaking them, reforming them, breaking them, reforming them until you get a very thermodynamically stable conformation. Another protein that helps these proteins achieve their proper tertiary and quaternary structure are chaperones. These are proteins that help other proteins fold properly. Now, chaperones were originally called heat shock proteins because we first discovered them becoming much more abundant in cells after they had been subjected to high temperatures and thus heat shock. But now we know that they actually assist protein conformation in many situations. It's not just heat shock. That's just the first place that we discover them. And there are three major families here. There is the heat shock 70 proteins, the HSP or heat shock 90, and the heat shock protein 100. Now each of these is named because of the approximate weight in kilodaltons of each of these proteins. Okay, so let's talk about protein quality control, just generally and in terms of quaternary structure. We have quite a few proteins, these are associated with heat shock proteins, which are known as binding proteins, or BIPs. These belong to the heat shock 70 family of chaperones. They're also known as the GRP70s, which these GRP coming from the name glucose regulated proteins, and they are 78 kilodaltons. And another of these protein quality control proteins is endoplasmin, which is an HSP90 family, and it's also known as GRP94, again, glucose regulated protein and 94 kilodalton size. So oftentimes we need to bring together separate polypeptides to form one functional protein. And if the structure is improper, BIPs or endoplasmin can bind to it, and this will lead to the destruction or reformation of that protein. BIP is also known as the master regulator of the rough endoplasmic reticulum. This prevents the clumping of the nascent polypeptides or the signal proteins or it signals proteins for destruction, so helping maintain the proper formation of these proteins. Okay, let's talk about a couple more chaperones, particularly Calnexin and Calreticulin. These are chaperones that use calcium as a cofactor. Now, improperly folded glycoproteins are going to get a terminal glucose residue added to their oligosaccharides. So they're going to get an extra glucose tagged on to the end of their oligosaccharides. Once that happens, Calnexin or Calreticulin can now bind and prevent the glycoprotein from leaving the endoplasmic reticulum for the Golgi. The Golgi is the next stop for most proteins after it leaves the endoplasmic reticulum. This will prevent it from going on. You're kind of stopping that protein in its path. And this will also signal it that's destined for degradation. We're going to break down that protein because it is improperly folded. Okay, let's talk about another pathway for breaking down and destroying these improperly folded proteins, or this can also happen to proteins that we simply do not want to use anymore. And this is the ubiquitin and proteasome pathway. So this is known as, one portion of this, or one version of this, is known as ERAD, or endoplasmic reticulum associated degradation. One of the steps in this process is known as molecular triage, which are steps to determine if a protein needs to be refolded or it needs to be just degraded. So let's first talk in this pathway about ubiquitin. Ubiquitin is a small protein. It's about 76 amino acids. And then we also have the 26S proteasome, which has two each of three different subunits. Have the cap, which functions in recognition of proteins. We have the alpha subunits, which are involved in unfolding the protein, and the beta subunits that are involved in breaking down the protein, or proteolysis. So, in this process, a protein that is to be destroyed is going to receive a polyubiquitin tail. Essentially, many ubiquitin, or several ubiquitin, are going to be added to the tail or to one end of that protein. And then degradation is actually going to occur in the cytosol and on the RER outer membrane, not inside the RER lumen. So it needs to get to the outside of the RER. So what happens is certain chaperones, particularly BIP and another one known as PDI, are going to direct them back to Sec61, Remember that Sec61 is part of the translocon that actually forms the channel that allows polypeptides into the cell. Well, in some cases, it will also allow polypeptides back out of the cell. And the ubiquitin is going to be added as the polypeptide leaves the RER. So, in this case, we find that a polypeptide is improperly folded. BIP and PDI are going to bind to it, direct it back to Sec61, and as it leaves the RER, ubiquitin is going to be added. So, here's what we have going on. We have a misfolded protein. We have a chaperone that's going to bind to it. It's going to come back to the RER membrane, and specifically to Sec61, where it gets transported outside. As it gets transported outside, we're going to get this polyubiquitin tail added to one end of this protein, in which case is going to then move on to be consumed by the proteasome, which is essentially going to surround it. There's a little hollow area in here in which the protein will be in there and be essentially degraded, and those amino acids then can be regurgitated, or not regurgitated, excuse me, recycled and used for other proteins. Okay, let's talk about another thing that can happen to or as a consequence of having misfolded proteins. And this is what's known as the unfolded protein response. Now, this particularly happens when we have a lot of misfolded or unfolded proteins in the RER. We have quite a few of them. We're getting an accumulation of these misfolded or unfolded proteins. Something kind of more major is going on. There's actually 3 sensors, membrane receptors, that can detect excessive amounts of unfolded or misfolded proteins in the RER lumen. These are PERK, ATF6, and IRE1. BAT and BAX, these 2 proteins that we have talked about before in apoptosis, control and modify RER1. And we'll come back to that in a minute. They're on the RER as well as associated with the mitochondria. So they're not just occurring on the mitochondria, they're also occurring in association with the endoplasmic reticulum. So, 3 events are going to happen in the unfolded protein response. The first is that in existing chaperones that are in the endoplasmic reticulum are going to be induced to enhance their activity. They're going to start up working extra hard. And this is going to be caused by the action of ATF6. Also, there's going to be a reduction in translation of new polypeptides. If you're making bad polypeptides that keep seeming to be broken, we don't want to make more. You don't want to keep just accumulating these really poorly made polypeptides for whatever reason. Whatever reason you happen to be making those, you don't want to continue that. And that is going to be accomplished by PERK. Also, we want to promote the transcription of more chaperone genes by sending signals to the nucleus to upregulate chaperones. So all 3 of the gene, or all 3 of the proteins that we talked about, are going to be doing that. If all these things don't work, so sometimes you can do all of this. You can start cranking up your chaperones. You can make more chaperones. You can cut back all the other translation that you're doing. And it still doesn't work. You are still accumulating unfolded proteins. Well, if this doesn't work, the first thing that can happen is we're going to destroy that rough endoplasmic reticulum by lysosomes. There could be something just fundamentally damaged about that RER, and so let's destroy it and make a new one. Alternatively, this could also lead to apoptosis. If this is a more widespread problem than just this being a bad RER, this entire cell might be doing some bad things, and we need to actually destroy that cell to preserve the whole organism. So those are the steps that will happen in the unfolded protein response. And essentially, this just sums up the actions of the rough endoplasmic reticulum. With the next lecture, we're going to come back and go on to the next organelle in the chain of polypeptide synthesis, which is going to be the Golgi.